Okay, first of all, I would like to thank the organizers for organizing this meeting and for inviting me. I am in this minority species of experimentalist in this meeting. And so I will be talking about the topic was already read out. This work was done in collaboration with these people. Arsalan and Sousa were students who performed most of the experiments. Serene helped us with all the molecular biology techniques and so on, and they are at Raman Research Institute. And Pearsons and Taba Singha, they are both at Institut Curie Paris, and they did the theoretical work. And then there are people who helped us with various aspects along the way. So the experiment is as follows, that you have a neuronal cell, that is a cell body. This is this long tube which is known as the axon, and it ends in a tip which is called the growth cone. These names are not so important. What we do is we come with a spherical particle. This is a bead, 1 micron in size. We trap it in an optical trap, which is just a hand to hold the bead. And we come and stick the bead to the membrane and we pull. And when we pull, what it does is that it pulls out a membrane tube out of the cell, out of the axon. Okay, so that is what is shown here. And here is a, so if you do that, as soon as you pull, you see that there is a sudden increase in force, and this force relaxes with time. And this is a viscoelastic-like process which causes this relaxation. And if you again pull, like these are the pulling steps, you apply more and more steps. For each step, there is a spike in force, and then it relaxes and comes to a stable value. And if you don't stretch or pull, then the force will remain at this constant value. Okay? And here is a video of the experiment. So that is the bead, this is the axon, and that is a tube which you pull out of the axon by doing this. So in our experiment, what we do is we pull it by a certain distance, that is you displace the trap from the axon by a certain distance, and you keep observing the bead and you ask what is the bead doing as a function of time. And when you do that, you get a time series which looks like this. Instead of, like I said, in most cells, like when people do these experiments, you see a constant force at steady state. But in this case, you don't see a constant force at steady state, but the force shows this kind of load and fail behavior, repeated load and fail cycles. And this will go on for tens of minutes if you keep observing the system. So our aim here is to understand why you get these load and fail kind of response. So you need to know what is happening inside the tube. So we imaged the components of the tube, and we found that this tube is filled with filaments which are called actin filaments. And these are very dynamic filaments. These are made of protein subunits, and they polymerize to form this sort of structure. And these filaments can add monomers at one end and thereby increase in length, or remove monomers and thereby decrease the length. Okay? So that is what I mean by being dynamic. So when we image what's happening in the tube, we see that what is seeing as bright is actin filament, which is fluorescently labeled. You see that once in a while these filaments retract, and then again they grow back, they go to the tip of the tube. This is where the tip of the tube is, and then again retract, again grow back, go back, again retract, and so on. So there is this growth and decay cycles which is happening inside the tube, the growth and decay of actin filaments. If you correlate these growth and decay with force using the optical trap measurements, you see that here there is no continuous bridge of actin. Okay, this is the base of the axon, and the bright spot is the bead which scatters the laser light. So what we are interested in, what happens between these two very bright regions. And you can see that here there is a dark patch, and slowly actin grows and it spans the entire length of the tube. And when it spans the entire length of the tube, the force starts building up. Okay? And the force collapses when the actin detaches and begins to retract. Okay? So these are the cycles. Okay? When the actin grows, reaches the tip of the tube, binds to the bead, pulls, and then suddenly there is a detachment event and the force collapses and comes back to the initial value. So this is interesting because normally when you have these kind of contractile force behavior, these are contraction of the tube. These are generated by what are known as molecular motors. So you can take systems of like single cell systems like sperm which is swimming. And if you look at the flagella of the sperm, you have this arrangement of filaments. These are microtubules, not actin in this case. And they're connected by these purple structures known as molecular motors. And these molecular motors can walk on these microtubules and generate contractile stresses. And these stresses causes bending of the tail, and that is what makes the sperm swim. Same in the case of muscle, irrespective of how big or small it is, that inside what you have are structures like this, where you have actin filaments in this case, and these are myosin molecular motors which attach to the actin filament. They walk on the actin filament, and thereby they pull this filament together, and this is how the muscle contracts. Okay? So these are forces generated by these kind of single molecular motor proteins, and what the energy for this comes from the consumption of ATP, adenosine triphosphate, which gets hydrolyzed. So if these sort of general force generation mechanisms are summarized in this recent review article which we have published. So what happens in the case of tube? So one could imagine that there are these sort of motor proteins in the tube, and these motor proteins are pulling on actin filaments, and they may be motor proteins sitting at the base, or they may be motor proteins distributed along the tube. And the easy argument would have been that, okay, these motor proteins are responsible for the pulling, and whenever these filaments detach from the tip, then you get a collapse in force. But we did a set of experiments. I will not go into the details of these experiments in the interest of time, but basically you can use various chemical agents and you can block these various motor proteins which are present in the cell. And we found that if you block these motor proteins, we still see this load and fail behavior. So molecular motors are not involved in this process. What is involved, however, is actin dynamics, actin polymerization and depolymerization. If you sort of inhibit that process, then you don't see the load and fail behavior at all. So the question is, if molecular motors are not involved, then what might be causing contractility in this? So we then came across this certain proteins called cofilin, and what these proteins do is these are actin binding proteins. So they come, and if you allow these proteins to come and bind to these actin filaments, they make some sort of allosteric changes to the protein conformation. Okay? And these allosteric changes are such that they over twist actin filaments. So the pitch of the actin filaments, if you remember the picture I showed, it's a double helix, and the pitch of the actin filament is decreased when these proteins are binding. So it generates torsional stress in this filament. So we asked ourselves is the question that can this sort of mechanisms generate stresses in these filaments and drive contractility? So we did various experiments. We fluorescently labeled this protein cofilin, and we found that sure enough these are there in the tube. And then there are various ways of inhibiting the binding of this protein to actin filaments. And when we do that, we see that the force generation goes down. These are competitive binding, so you're not inhibiting 100%, but you're reducing the efficiency of this protein, its ability to bind to actin. Then you see that there is a reduction in the maximum force it can generate, and also you see these very long-lived events which suggest that forces are generated for a long time, and the forces are not large enough for the actin to detach. So we then developed a simple model. The model is very, very simple, that you have these actin filaments. They are attached to the tip of the tube to the bead. K-trap is the bead, and you allow these cofilin to bind stochastically. They are K on K off of that. And these tubes can, the tip link can also break stochastically. It has a binding, unbinding kinetics. Okay? And every time a cofilin molecule binds to one of the actin monomers, what it does is it generates a small deformation of the actin monomer, and this stress results in a shrinkage of the monomer by some length delta. Okay? So you can write down equations for this, and then you can simulate these responses. And we see that these are what is shown in red are the experimentally seen shapes of these force events, and these are the simulated ones. And you see that all the features which we see in experiments can be reproduced in the simulations as well. Okay? So what happens in this case, for example, is there is a partial drop. This happens when one of the filaments detach, while the other filament is not yet saturated with cofilin. So the other filament continues to generate a force. And you get these saturation events if all the filaments are completely saturated with cofilin, and the detachment even occurs after all filaments are saturated. We can also compare the statistics of these force time series, and we also see that what is shown in red are the experimental data, and what is shown in black are the data we get in from simulation. So this is a pictorial summary of what I just described, that actin filaments grow inside this tube, this membrane tube, And when they polymerize all the way to the tip, this molecule called cofilin start decorating the filaments. And when cofilin decorates, there is a stress which is generated in the filament, and when the stress is large enough, these filaments disintegrate and they depolymerize, and then the whole cycle repeats again. So this is a kind of novel mechanism for contractile force generation in actin filaments, and this acts like a linear cyclic actuator. One might wonder, where does the energy come from? The energy comes because this polymerization, depolymerization process involves ATP hydrolysis. Otherwise, you cannot drive this cycle. And this work is summarized on this article available on bioRxiv, and with that I thank you.
In that first slide, you had this strain applied, and then this response which is spiked, and then
yes.
So if I had applied a constant strain at the time scale which is comparable to that spike,
right?
Then what's the response? Will the response change?
Yeah. So if you mean a constant strain rate or constant strain?
Like you are applying.
So here, what is applying is a constant strain. Yeah. So you apply a step and then you maintain the strain constant.
Yes.
Yeah.
The strain is constant.
Yes.
If the rate of change of strain is larger, then as compared to the spike time, then how does the system respond?
I would imagine that this force would just keep increasing with time, because I'm just without the spikes.
And the other question is, suppose I preload this with some cyclic loading for some, and then I repeat the experiment. Will the response have some memory dependency?
We have not checked it. Okay.
So the fibrils and so on.
So one of the models, in this paper, we give a model to, so this relaxation is not an exponential relaxation. Okay? So we gave a model where we could explain this sort of relaxation behavior. The details I have not plotted I have not enlarged this plot, but to get the proper fit of this, you need to consider migration of components. So as soon as you pull, the tube composition is different from the composition of the axonal membrane. And this neck forms some sort of a barrier to diffusion. Okay? So things have to diffuse across this barrier into the tube during this relaxation process. So if you consider that kind of process, then what you said might be there. There may be memory effects coming because now there is another time scale involved, which is for this migration to happen. But we have not checked that as well. Something which is interesting to check.
Okay, so what I understood is like you have lots of filaments which connect with the
a few filaments.
A few filaments, right? And so you get the jumps when all the filaments break?
No,
or,
no.
or
they're not synchronized.
So whenever even one filament...
exactly. So I showed these events where there are these... okay, for example, okay, let me go back to this. So you see that here there is a discontinuous change in slope. Okay, this happens when one of the filaments has detached, but the remaining filaments are not yet saturated. So they continue to pull on the bead, but with a smaller force now.
Like, but the big jump that you see...
The big jump is, this is a catastrophic collapse of all filaments simultaneously.
That's all, but okay.
But you do see cases where it's not a complete jump, but it jumps partially, pauses here, and then jumps and so on.
Okay,
so,
and then they start polymerizing again and connecting.
Exactly. That's the full cycle. And the polymerization requires a hydrolysis of ATP. This time distribution this is randomly distributed. The dead time, right, between two events. Yeah, so when a filament collapses, then the load is now shared by the remaining filaments. So the probability of the remaining filaments detaching becomes higher. Is that what you're saying? You mean control the force on the bead and gives us some sort of feedback?
Yes. If there are no more questions, let's thank the speaker.